Thank you, Diane. So to wrap up this symposium, I thought I'd tell you a little bit about some of our work in southern Zambia through the International Centers of Excellence for Malaria Research funded by the National Institutes of Allergy and Infectious Diseases. And what I want to do is try to bring together a number of the strands that have come out during the day. And we've certainly talked a lot about Zambia, and I'm, again, very delighted that Dr. Chizema is here with us today. So the Southern and Central Africa ICEMR is a large collaboration of institutions, both here in the United States and in Zambia, as well as Zimbabwe, and South Africa as well. So just a little note about the International Centers of Excellence for Malaria Research. These were initially funded in 2010 for 7 years. And then there was a recompetition extending from 2017 to 2024. We have been very fortunate to have received what will be 14 years of NIH funding for a single project. And this slide shows the distribution of the current set of ICEMRs around the world. And these are really multidisciplinary projects that are really aimed to kind of tackle and understand malaria epidemiology, entomology, parasite genomics, and control efforts. So the Southern and Central Africa ICEMR has 2 sites in Zambia, and one in southern Zambia, that's based at Macha Research Trust. And as we've talked about, this is an area in southern Zambia where malaria control has been effective, and we've seen a decline in parasite prevalence and disease burden. Our second site is in the north, in Luapula province in Chilenge district, on the border with the Democratic Republic of Congo. Our third main site is in eastern Zimbabwe. I'm not going to talk about that today, on the border with Mozambique. And then we've done some work across the border in the Democratic Republic of Congo in Haut-Katanga province. But I'm going to focus today's brief talk on work in southern Zambia, where we've been trying to assess some of the barriers to malaria elimination. And again, I'm going to try to draw on a number of the themes that came out of the talks here. So the main question in a big picture sense is, how is low-level transmission sustained, and how do we move and achieve and sustain malaria elimination in such a setting? And you've seen some of these graphs earlier. On the left-hand side is actually modeling from Imperial College. Dr. Chizema showed this, the decline in malaria in annual cases, actually, over the past 15 to 17 years. You can see that there was a fairly dramatic decline in the first part of the decade of the century, and then pretty stable over the past number of years. Dr. Chizema was kind enough to share the data from the map from the most recent malaria indicator survey in Zambia, and you can see that and actually this relates to the prior discussion, actually Rick's discussion of Senegal, where you have great heterogeneity in transmission within a country. So we're working as the Southern Africa ICEMR in southern province here. You can see by microscopy, actually the prevalence was 0. We know that malaria has not been eliminated there, but it was very low in the malaria indicator survey. And then obviously very high in Luapula province, which is where Chilenge is. So I'm going to talk about what's happening here through our studies. And the way I want to frame this is by talking about some measures or metrics of progress toward elimination, some of the challenges, some of the issues around sustaining elimination, and then documenting elimination. So we have a number of different ways that we've measured the decline in malaria in this area. Dr. Phil Tuma, who's in the audience, has collected pediatric hospital data over a long period of time, going back to 1999. And you can see this decline in the number of hospitalizations of children here occurring around 2004, 2005. So the first point is this dramatic decline. But what I want to focus on in our studies is this kind of maintained low level of pediatric hospitalizations for severe malaria. We started our work during active case surveillance, where we would randomly select households from the surrounding community using satellite imagery as a sampling frame, and then measure parasite prevalence both by RTAT and using a PCR. Again, you can see this kind of decline in parasite prevalence now through active case detection. We actually stopped doing that in 2013 because the parasite prevalence was so low. But I want to highlight this kind of background prevalence of about 1%. We also do use passive case surveillance as the National Malaria Elimination Program does through rural health center reporting. This is that kind of, this is for 14 rural health centers in the surrounding area. There are weekly RTAT positive cases. We did see, well, first let me point out, this is kind of really low historically for numbers of cases in this area. We did see a slight uptick in 2016. But again, the point here is that there's been this low level of transmission of malaria parasites in this study area. So what are some of the challenges to elimination? I'm not going to talk about all of these. We and other studies, I know the Malawi ICEMR and Miriam Laufer was here, documented the highest parasite and gametocyte prevalence in school-age children. There are certainly limitations of rapid diagnostic tests in this setting, in this low transmission setting. As many people know, the sensitivity can be very low. There's some transmission of non-falciparum malaria, particularly Plasmodium malariae, and HRP2 deletions, at least a hint of those. I'll talk a little bit more about that. We've heard about the step D or component D of the strategy that was used in Zambia and is recommended by the World Health Organization. We've been evaluating this reactive test and treat strategy. I'll tell you more about that. Toon talked more about the infected asymptomatic reservoir. And then one thing we haven't talked too much about today is the role of secondary vectors and residual transmission. There's been some discussion of that. So just very briefly, this is complex data. This was a study led by Tamaki Kobayashi looking for PFHRP2 deletions in our high transmission setting in Chilenge and in the low transmission setting in Choma. Just to cut to the chase, in Chilenge district, out of 28 samples that were RTAT negative but PCR positive, we did not find any evidence of PFHRP2 or double deletions in that area. But there were 3 out of 8 samples in the low transmission setting where we could not detect the PFHRP2 gene, despite being able to detect PFHRP3. Now, these are very low levels of parasitemia, so the big caveat here is that it's possible that we just had insufficient DNA to detect this gene, but this was kind of a hint of detecting PFHRP2 deletions in the low transmission setting. You've seen this figure before. I mentioned this earlier, the component D or step D as it was called in Zambia. The way this worked in Southern Province was that when an index case was identified at a health facility or by a community health worker, was shown to be RTAT positive, that healthcare worker went to the index case within a week and tested and treated everyone within 140 meters radius of that household. So we've been doing a lot of work to try to understand some of the challenges to doing reactive test and treat in this setting and some of the limitations. And so what we did as a study, for study procedures, and we started in January 2015, we accompanied the community health worker on the reactive test and treat strategy, but we extended the radius to 250 meters. We collected a dry blood spot for qPCR, and then in 2016 we initiated follow-up visits at 30 and 90 days. So I'm just going to tell you very briefly the results of some of these evaluations. Our first evaluation looked at parasite prevalence by both rapid diagnostic tests and by PCR within the index case households, within 140 meters, and within 250 meter radius. A couple points. The rapid diagnostic test, and Toon alluded to this, was about 50% sensitive compared to the qPCR. So with the PCR, we were detecting about twice as many cases. The second point is that most infected individuals were within the index case household. And once you went outside the index case household, the parasite prevalence was pretty constant, whether you were within 140 meters or within 250 meters of the index case household. And again, it was approximately double. So this is about that 1% background prevalence that I alluded to earlier when I spoke about the active case detection. So we think in this setting, and at this time point, about 1% of the population has low-level parasitemia, and these are, by far, the vast majority of these are asymptomatic and afebrile. So based on this, one could argue, I think, that perhaps a more efficient strategy would be to do a focal drug administration within the index case household and not use an RTT and not try to extend that radius beyond the index case household. Now, in a study led by Kelly Searle looking at the follow-up visits, this shows over three transmission seasons. What you have here is the initial visit in red, the 30-day follow-up visit in blue, and the 90-day follow-up visit in green for within the index case household and within these two different radii. And the bottom line point here, the numbers are very low, and this is one of the challenges of studying malaria transmission in a very low transmission setting. But the main takeaway here, I think, is that the reactive test and treat strategy did not eliminate malaria within either the index case households or within any of the radii. There were still infected individuals at the follow-up visits at 30 and 90 days. And I'm going to talk a little bit about the use of genotyping to try to sort this out. We're still working through some of this. But the reactive test and treat did not achieve elimination in this setting. David, just to link back to David's talk, David showed this figure, again, Kelly's study, where we asked participants to wear a GPS logger and looked at very fine spatial movement. This also links to Amy's discussion about population movement. What GPS loggers give you is very high resolution spatial patterns of movement, but on a smaller number of individuals. You can't do this on a large scale, and Amy pointed that out. One thing that we noticed, though, that we thought was interesting and has some implication for elimination in this setting are the seasonal movement patterns. And so in the rainy season up here in December and February, the long-distance movements was very restricted. People tended to stay around their homes. We think this is probably because they're tending to farms and because perhaps of washed-out roads. And then in the dry season, we started to see longer movement patterns. And it was actually probably in April where we had the greatest chance of someone traveling far enough where they might get infected and be able to bring parasites back. We've also used genotyping to try to capture this difference or to address this question of whether the asymptomatic reservoir is really contributing to ongoing transmission. And this study has its limitations. This was also led by Kelly. But basically, using a 24-SNP barcode, compared parasite genotypes from individuals who were presenting with symptomatic disease through passive case detection, and these are indicated in red here, and those captured by active case detection in the community. This was largely that asymptomatic reservoir. And I don't want to overstate the findings here, but in general, the parasites identified through active case detection in blue were separate population from those detected through passive case detection. Now, we certainly did not capture all of the infections in this population, so we may be missing areas of overlap between these two parasite populations. But in general, this suggested that the asymptomatic reservoir in this setting was not contributing substantially to the symptomatic cases, but again, with that caveat. Julia Pringle has also used parasite genotyping methods, and actually this was in collaboration with Brian Greenhouse, to using their microsatellite markers, looking at, in the reactive case detection, genetic relatedness. And again, this is a complex data set, but the bottom line here is that the most related parasites were those that were detected closest in distance and closest in time. This suggests some ongoing local transmission of highly genetically related parasites. But this gives one example, kind of coming back to Brian's earlier talk, of how one might be able to use genotyping methods to get a better picture and a better understanding of local transmission and imported parasites. The last challenge I want to talk about are some of the secondary vectors and outdoor transmission. And this is work led by our entomologists, both here and in Zambia. And I just want to highlight in the outdoor collections this predominance of Anopheles squamosus. And when we looked at these more carefully, one, we noted that there were two clades of squamosus, but these specimens identified in red actually were captured Anopheles squamosus that had Plasmodium falciparum. This was a previously unknown vector in this area, and it just shows that as transmission goes down and you start looking more carefully for vectors, you can identify secondary vectors that may be important in maintaining transmission. And so we've got squamosus that is perhaps largely feeding outside, and this is not going to be affected by our vector control strategies. So those are some of the challenges. Linking back to some of our earlier discussions throughout the day, I want to talk about some of the methods of achieving and sustaining elimination. Some of this is ongoing work, and I'm not going to talk about everything, but Matt Ippolito took a look at different housing structures in this area, and perhaps not surprisingly, we were talking about this yesterday, this has been known for 100 years, but if you have better housing structure, either cement walls or brick, you're at much lower risk of having RDT confirmed malaria in those households. And we know PMI, one of the efforts that PMI is leading is looking at improving housing structure in some of these areas as an additional malaria control strategy. We've also been thinking about ways to make reactive test and treat more efficient. And so the general strategy is to use a circular radius around the household, but that can be very convenient for community health workers, but may not be the most efficient strategy. So we're using satellite imagery to try to identify ecological features that might be able to be used to identify high-risk secondary households. And this was an analysis led by a student, Fiona Bonderkun. I'm showing images of where she identified animal pens within a household. That was one of the covariates that she looked at. But it turns out in the analysis, it was proximity to certain category streams, certain size streams that were a risk factor. So just to put it out there, one strategy for reactive test and treat, instead of doing a circular radius, might be to identify the nearest body of water or stream and go along that, and one might identify more secondary cases. Brian also talked about serology. This was work led by Tamaki, but in collaboration with Phil Feldner's group, using a protein microarray with 500 different P. falciparum antigens and measuring the antibody responses to those. Here we're just focusing on the top 30 antigens across our 3 ICEMR sites. And I just want to point out, not unexpectedly, that you see very different age-specific seroprevalence curves here. These are the top 30 antigens. We've got age categories on the x-axis. This is the antibody concentration. But what I want to point out, and Brian so this is our low transmission setting. This is our high transmission setting in Chilenge, and this is our intermediate transmission setting in Zimbabwe. And I think the striking differences here is that the young children in Choma district basically were seronegative. The children under 5 were seronegative. And so it's using serology as a marker of malaria elimination or the cessation of transmission could be very powerful in these low transmission settings. Just in contrast, in our high transmission setting, young children under 5 were almost had equivalent seroprevalence as the adults. We have one thing that we're doing now. This is a project led by Jessica Shu, is to set up, somewhat modeled after the Uganda ICEMR that Brian talked about, but to set up an intensively followed longitudinal cohort where we're following individuals every month, getting travel history, collecting a blood sample. And what we're hoping to be able to do, working with Brian, is to identify, using parasite genotyping methods, transmission networks, and really being able to quantify the amount of local transmission in this very small area within southern province Zambia compared to importation. So this study really builds on Brian's talk, where we're using these newer tools of parasite genotyping and creating transmission networks, and then also because we're hoping to identify all incident infections, to be able to use serology to identify specific Plasmodium falciparum antigens with different half-lives so that we can kind of capture that window of time of infection as Brian was talking about earlier. We're also have a collaboration with Toon looking at the infectiousness studies here and hoping if we can pull it off, get the assay working, see if we can identify whether some of these individuals who have asymptomatic infection are actually infectious to mosquitoes, and then continuing to explore the role of secondary vectors. So just in summary, what I wanted to do was kind of give you a picture of a set of studies that the Southern and Central Africa ICEMR is doing in this low transmission setting in Zambia, trying to pull on some of the themes that we've talked about today. We can see that low transmission has been sustained in this area, but we haven't been able to move toward elimination. There are a number of challenges to elimination in this setting, and using these newer tools, parasite genotyping and serology in this low transmission setting to try to better understand the transmission dynamics. This is the work of a very large group of collaborations that we have in the Southern and Central Africa ICEMR. With our partners at Macha Research Trust, which is based in southern Zambia, TDRC, they're our main partners for our project in Chilenge, the Protestant University for our work in the DRC. We have 2 groups that we collaborate with in Zimbabwe, the UNC and University of Massachusetts. Actually, I need to change that to Brown. Jeff moved to Brown. That's our parasite genomics core that's helping us with our parasite genomics, and then a large team here. I just want to end by acknowledging the support from the National Institute of Allergy and Infectious Diseases and the Bloomberg philanthropies that support these studies. Thank you.
Thank you, Bill, for a strong finish. Are there questions? Lou.
Tom.
Yeah.
Oh, sorry, Lou's over there. Yeah.
I want to know if when they came back 90 days later and there were new infections, were these different people? Because if they were, I have a suggestion. So why don't you tell me the answer?
Yeah.
There were new people.
Some of them are different people and some are the same, but they're also similar related parasites.
But that would make me think, if
Yes.
they were different people,
Yeah.
that I just treat Massive Drug Administration rather than try
Right.
to mess around with.
Right. No, I agree with you, Lou. I think, in this type of setting, and it's one of the big challenges of reactive test and treat, if you're relying on a rapid diagnostic test, it's not sensitive enough to detect those low levels of parasitemia.
Thank you. Marvelous talk. I really enjoyed that. A question is, as you were detecting different populations in the, or essentially the asymptomatics and the symptomatic patients, what is your leading hypothesis for that? The passive and active detection distinguish, I think it goes in parallel.
Right. So Tom's asking about, right, this separation of the parasite genotypes between the active and passively connected individuals. And, you know, I think there are a couple of explanations there. One is that a large proportion of the symptomatic individuals may have acquired their infection elsewhere. But I also think that and I think Julia's analysis with the microsatellite shows that there is some ongoing local transmission. And so I don't have the definitive answer to your question, but we're hoping that our intensively followed longitudinal cohort will answer that question.
And a quick second question, if I may, and it may be related to your answer here, is the serologies we're showing in the younger children, you didn't see much, and then, of course, the older ones. And you think that's chronic exposure?
Yes.
And you have variation in those antigens that may indicate different parasite populations. So, of course, a fresh parasite infection would lead to symptoms. Okay, thank you very much.
What we're working on now is, so that the analysis I just showed you looked at the 30 antigens that gave the strongest signal. We're going to start looking. We may see more interesting patterns actually as we look for some of those lower level antibodies. Yeah, Dave.
Hi, David Smith from the University of Washington. If you plot out the locally or the ones you found by active surveillance, if you plot them out spatially, do they cluster spatially as well? The blue ones on your, you know, the ones that separated out that you found by active surveillance.
Were they spatially?
Yeah. So what we have done, Dave, is we've taken those spatial patterns and generated risk maps, and so it's not homogeneous. We've also done hotspot detection with some of the passively detected. So I guess my answer to you is that there's not. There's spatial heterogeneity. That's how I would frame the active case detection. Yeah.